We will now extend the single layer that we studied to situations where there can be multiple layers together. We want to know the extent of mass diffusion through multiple layers. So there can be many situations where diffusion is occurring through multiple layers, where each layer has a different property. So like here, each layer has a different property. For example, here we have moisture transport through 3 different clothing layers. In another example, we have urea transport through first convection, then diffusion through a membrane, and then convection again in a dialysis membrane. This is the one we will do in more detail. And yet another example, the drug from the reservoir, the active component would be diffusing through multiple layers of drug coating. All of these problems can be thought of diffusion through multiple layers together, and they can be conceptually seen as something like this, where this diffusion is happening through multiple layers in series. And so what we are interested in is how do we get the concentration profile as the species diffuses through, and how much is the mass flow. This is a hollow fiber dialyzer. Blood comes in, and let me show. So the blood comes in through here and goes through these many fibers, which are hollow tubes. Hollow tubes. So many thousands, tens of thousands of hollow tubes through which the blood flows, and then the blood goes out here. Now outside of the fiber is the dialyzing fluid, the solution inlet, which is somewhat of a clear solution. It flows in and flows over the outside of these hollow tubes that are fibers, and then it goes out. The solution goes out. So blood is inside the tube, inside these little hollow tubes, and clear solution is outside the hollow tube. We now look at one tube of this hollow fiber dialyzer, or one fiber in detail, with the blood flowing through the tube and the dialyzing fluid flowing outside the tube. Purpose here is to have urea that is high in concentration to go out of the tube, and it will do so because the urea concentration is high here and outside is clear fluid, so urea concentration should be low. So then the diffusion should occur this way, and that's what we want to study. There are other transport happening in this dialyzer, but we are not interested in those for now. Now note that even though there is only one membrane, so it looks like there's only one layer, so why are we talking about multiple resistances? So if you think of the urea it first has to go from the interior points to the surface, and then it has to diffuse through the membrane, and then it has to go out. So there are really three different resistances for the urea, from between the high concentration in the blood and low concentration in the dialyzing fluid. We will now zoom in this wall, and we will see this one. It shows that we have higher concentration in the blood and lowest concentration in the faraway dialyzing fluid, and there is a concentration gradient that forms all along from the high to the low. Now, so using this problem as a background, we are going to come up with a way to solve multiple layers. So in this case, we have resistance 1, resistance 2, and resistance 3. So it's 3 layers. So we're going to see how we can use this problem as a prop to study multiple layers.